Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Revinta Nur Aulia Rochly and with study number 4411420010. I'm from Biology Class 2A. On this occasion, I will present a scientific journal with the title Identification of Morphology Characteristic of Molds and Yeast that Role in the Fermentation Process of Food Products. In the Bird Journal. Author is me, Revinta Nur Aulia Rochly, and institution Biology Study Program, Faculty of Mathematics and Natural Science, Semarang State University, Semarang, Indonesia. This journal has 5 parts. The first, introduction. Second, material and methods. Third, result and discussion. Fourth, conclusion, and the last, reference cites. The first part, introduction. What is fungi? How to identify the morphology? Fungi are the type of eukaryote organisms that cannot photosynthesis because they do not have chlorophyll. Some are unicellular and multicellular, and can reproduce sexually or asexually. To identify the morphology can be fungal staining. The study objective is to determine the morphology of yeast and molds that play a role in food fermentation. And theoretical basis, fungi can be divided into 3 types, namely yeast, mold, and mushroom. Mold is a multicellular fungus that has filament and has a fibrous shape. If it grows on food, so it is easy to see. While yeast is a unicellular fungus that can live in various places. Second part, material and methods. The writing is using descriptive methods. Second part, material and methods. The writing is using descriptive methods or literature study methods. That is read through, compare, analysis, and sort literatures in order to identify the essential attributes of material. Research on molds and yeast cells was carried out using the staining methods. Staining of molds with LPCB solution or sterile aqueous, while yeast with 0.1% methylene blue solution. The fungi that use as the pure culture of the mold is Rhizopus oryzae, Penicillium species, and Aspergillus oryzae. And pure cultures of the yeast such is Saccharomyces cerevisiae and Candida humilis. Part result and discussion. Part is the first, Rhizopus oryzae. Rhizopus oryzae has hyphae in the form of branching rhizoids. The hyphae are not insulates or choanocytic. There is a sporangium that produces spherical spores. There are stolons which are hyphae between the sporangiophores. These stolon looks brown. Sporangiophores grow upward. This fungus is used in fermenting soybeans into tempe. Second, Penicillium aspect. Penicillium species. The fungus have insulated hyphae that form conidium, where this conidium has branched called pileates. This fungus also has conidiophores that produce conidia, which are single cells produced by pileates. This part of this pileates is covered by sterigma. The fungus Penicillium species it is used to produce antibiotics and give blue cheese its tasty and aroma. Next, Aspergillus odyssey. Microscopically, Aspergillus odyssey has insulated hyphae. There are conidiophores that grow freely and irregularly. Conidiophores contain conidia. Has conidia whose head is bright greenish yellow and there are vesicles. The fungus also has more sterigma and pileates. This fungus is used in the manufacture of soy sauce and tauco. Next, Saccharomyces cerevisiae. This is the type of yeast. Microscopically, looks ovoid to elliptical in shape. This fungi is commonly used in making bread, alcohol, beverage, and tapai. And the last, Candida humilis. Microscopically, this fungal cells are oval in shape. This fungi is mostly found in sourdough, starting to give it tasty and aroma. Part conclusion. Mold are composed of branching hyphae or mesicle, called mesicle. And yeast has a cell shape that is mostly round or oval. Fermented products that are generally produced by molds are such as tempeh, cheese, soy sauce, and tauco. While for yeast, the fermented products are bread and alcoholic beverage. And the last part, preference cited. Sorry if there were mistakes and thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.